Welcome everyone to a short video about the EVOS microscopes present in the practical lab, which will be used during these practicals to take pictures and observe your cells. So the EVOS microscope is a little bit different from a normal microscope because the light enters from below and the lens is made in such a way that it takes the few millimeters of the bottom of the plate into consideration. So that way you can focus on the cells attached to the bottom of the plate. So what we do is it has this protective box, which when you use fluorescence, this box has to be on top of it, so the light around us doesn't affect the measurements. So I can put my cells in here. We can see the different magnifications in here, and then I put the cells here and I put the box on top again. So then we have different knobs and wheels that we can turn. First we turn the light on, and then we have an overview of the cells. We have these different knobs on the sides, and what you can do with them is you can adjust the image. So what you're actually doing is moving the plate where our cells are up and down to put the cells into focus. So we move it a little bit around, and then once we have a good image, what we can do actually is with this wheel at the bottom, we can turn it around and turn the magnification. Then we get our cells, we can see them better, and then again we turn this wheel here to really be able to focus on our cells. And then if we want to look at another part of the plate, that's what these two knobs are for. With this one we can go from left to right. With this one on the knob on the left we can go up and down. And that way we can look through our plate and look what our cells look like. So normally we start with the lowest magnification, just to search for good spots where there's a good amount of cells growing. And once we have found a good enough spot, then we can zoom in with the wheel below. Then we bring the cells into focus again. And this way we can have more details about the cells. These are U2OS cells. And we can see things like, are they stressed? Are they looking okay? Are they happily dividing? That's all things that you can see here when you zoom in more on the cells. So then you can also take pictures with this microscope, and you can also take fluorescent images with this microscope, and you can do that with a small handle back here, where we can go to red fluorescence, green fluorescence, and then blue fluorescence. And it has a very specific order. If you want to image different fluorescent channels, you're supposed to first image the red fluorescent channel, then the green fluorescent channel, and then the blue fluorescent channel. And that has a specific reason, and it's called bleed through. So if you start on the blue channel and you have fluorescence there, and then you go to the green channel, you're going to have some residual fluorescence bleeding through from the blue channel into your green channel. So you will have sort of false positives, where you will think, oh, this is green fluorescent, but it's actually fluorescent from the blue that is still present. So the appropriate order is to go from transparent light, then red fluorescence, green, and then blue. And you can actually take pictures with this capture button. You take one picture, then you go to, for example, the red fluorescence, take another picture, and then the cool thing is that you can actually also overlay the different colors that you take with each other. Right now I don't have any fluorescence, but that's something that can be done. So I hope you're looking forward to that, to making a lot of pictures and to see what overlaps with each other, what fluorescence, and what it actually means. Good luck.